Hey everybody, welcome to Supplementary video 10.3, 10.7 to 10.9. So over the course of the dual credit portion of the muscular system, our Unit 4, most of the information is already covered in the high school portion of the class. So we're actually only going to have 2 videos for the dual credit class here. And we're going to be kind of jumping around, just trying to hit on some of the things a little more in depth or adding in to the high school portion of this class. So that's why these sections are a little bit different and how we're jumping around. This will cover 10.3 and then 10.7 to 10.9. So the first thing we're going to cover is creatine phosphate metabolism. And so your cells need to use ATP as their source of energy, but for your muscle cells, they can kind of create a way to store more ATP because it's not the ATP per se that's providing the energy necessary for contraction. It's the energy in between phosphate bonds on those molecules. So ATP stands for adenosine triphosphate. So there are 3 phosphate molecules, and they have a very high energy bond in between them, especially in between that second and third phosphate. It's very unstable, very high energy. And so when a muscle is at rest and doesn't need to be using very much ATP, it's going to take ATP and transfer that third phosphate to creatine to form a molecule known as creatine phosphate. And that's going to allow the cell, it's going to free up space for that cell to create more ATP. And then when your body does need the energy provided by ATP, that creatine phosphate will transfer that phosphate back onto ADP, or adenosine diphosphate, which only has 2 phosphate molecules. It transfers that phosphate over to ADP to form ATP with the help of an enzyme known as creatine kinase, and then that ATP is available for that muscle to use for contraction. So creatine phosphate is what your muscle cells like to use, or it likes to store phosphate molecules in that way. And it's going to provide approximately 15 seconds worth of energy for a muscle contraction. You think if you go much longer than 15 seconds, a lot of those muscles, if they're fully contracted for those full 15 seconds, start to get very fatigued. And they start having to use other metabolic pathways in order to acquire energy. So here you can see the diagram going from ATP and creatine, that phosphate molecule getting transferred to creatine to form creatine phosphate, and leaving ATP to become ADP. Remember, this goes from 3 phosphate molecules to 2 phosphate molecules because that third one is located on creatine phosphate. And then with the help of that enzyme, creatine kinase, it goes back to creatine and you have your ATP available. The next thing we'll talk about is histology. So we'll look at cardiac muscle tissue here. Remember that cardiac cells have intercalated discs that are going to allow the heart to pump as a unit. And you need your heart to pump as a unit. You don't want it pumping in random ways because you're going to get all out of sorts and blood might be going the wrong direction. And so the cardiac tissue is able to contract as a unit through 2 structures known as gap junctions and desmosomes. So gap junctions are going to be channels in between adjacent cardiac cells. And what those junctions do is they allow the current, that electrical current from the depolarization of those the muscle cells to travel from one cell to the next. And so instead of having to have that current basically be restarted in between each cell, that current is able to flow through all those cells very quickly. And so it creates electrically connected contractions known as a syncytium. Syncytium is how you pronounce that. Desmosomes are going to anchor the edges of cardiac cells together so that they don't pull apart. Obviously, with your heart, there's lots and lots of stress from all those constant contractions. Desmosomes are going to make sure that your heart tissue stays together. So here you can see those gap junctions allowing all of those, or allowing that electric current to pass back and forth, and those desmosomes that are going to be holding those cells together. Smooth muscle, looking at the histology of smooth muscle. Smooth muscle does not have striations. We cover that in the high school portion of the class, but it still does contain actin and myosin filaments that are necessary for contraction. But instead of being arranged in bands like they are, which gives us the striped or the striated appearance of skeletal muscle tissue and cardiac muscle tissue, those actin and myosin filaments are a little more irregularly anchored, and they're going to be anchored to what's known as a dense body instead of the Z disc, which we see in cardiac and skeletal muscle tissue. Smooth muscle also does not have troponin, so it doesn't have that troponin tropomyosin complex that's blocking those binding sites. So instead, you're going to have calmodulin regulating that cross bridge that forms between actin and myosin. And so calcium ions in skeletal muscle cells and cardiac muscle cells, they're going to bond to that troponin tropomyosin complex. And in smooth muscle, those calcium ions are going to bind to calmodulin, which is going to activate myosin kinase, which activates myosin and allows it to extend and form that cross bridge with actin. In smooth muscle, you also have what's known as latch bridges, which allow certain subsets of myosin heads to remain attached to actin for very, very long periods of time without the need to spend ATP, which is really important to maintain at least some muscle tone in certain tissues. So for example, smooth muscle is any of your visceral organs. So that's like parts of your digestive system, parts of your blood vessels and things like that. So especially when we think about blood vessels, it's really important to maintain some level of tone in those blood vessels to keep your blood pressure at the correct level. If you lose that tone, your blood pressure drops and then blood stops flowing. Smooth muscle cells also have these bulges known as varicosities. They're going to contain neurotransmitters and are responsible for stimulating smooth muscle contraction. Because with smooth muscle, it is involuntarily controlled, not like your skeletal muscle cells where you can think about and tell a muscle to contract. Here, smooth muscle cells are going to be contracting on their own without you even needing to think about it. So you can see these dense bodies that actin and myosin are attached to, and then they're going to be pulling on those dense bodies, kind of creating this irregular, weird, bulgy football shape, if you will, in a contracted smooth muscle cell. Okay, and then last thing we'll look at here is the development and regeneration of muscle tissue. So mesodermal cells, remember mesoderm is the middle layer in the original layers of tissue that you have as you're developing. Those mesodermal cells that are right next to the neural tube are going to form somites. They're basically different blocks of cells. And those somites are going to give rise to myoblasts. Remember, myo meaning muscle, and eventually will make the skeletal muscles of the torso. They do not contribute to the skeletal muscles of the head or of the limbs. Those are going to arise out of just the general mesoderm. Myoblasts are basically muscle stem cells, if you want to think about them like that. They're going to migrate to different areas of the developing embryo, and they're going to fuse to form a myotube. And this is going to be the reason that skeletal muscle cells are multinucleated. So they start out as these myoblasts, these muscle stem cells that all fuse together. Well, when they fuse together, you still have all of their nuclei and everything within them. And so you're going to get multiple nuclei because those cells, original cells, all fuse together. Satellite cells are a different type of muscle stem cell. They're going to be responsible for protein synthesis, so actin and myosin and things like that, to help grow and repair muscles. You also have pericytes, which are a third type of muscle stem cell. They're going to be responsible for regenerating smooth muscle cells. Your smooth muscle cells do a little bit better job of regenerating when they have any sort of damage. So like you're able to get blood flow through angiogenesis pretty quickly back to areas that are damaged compared to skeletal muscle cells or cardiac muscle cells, which struggle a little bit more with regeneration. And in fact, muscles that sustain damage, especially in like the cardiac muscle tissue, they're going to replace that tissue with scar tissue if they don't have these pericytes. And when they have this scar tissue, those muscles are going to find it very difficult to contract because that scar tissue is not muscle tissue. And so it's not excitable, it's not extensible, it's not very elastic. And so it can't contract at all. Okay. So that, again, is one of our 2 supplementary videos for the dual credit portion of this class. If you have any questions on any of this stuff, let me know. You're also able to look at the textbook at those sections, 10.3, 10.7, 10.8, 10.9, to help yourself understand if you aren't able to. Contact me.